Okay, so talking about the agranulocytes, they are of two types. We have lymphocyte and monocyte. Now, lymphocyte, as you see, has a large nucleus. Will you remember that? Lymphocyte, large nucleus. Lymphocyte, large nucleus. LL. And the nucleus is so large that it takes up most of the space, and you can see a thin halo of cytoplasm on the periphery. Lymphocyte is responsible for producing antibodies. The next one is monocyte, and monocyte has a dented nucleus or a kidney bean shaped nucleus. Don't get confused. Monocyte does not have a bilobed nucleus. So if you look at basophil and eosinophil, they have a prominent bilobed nucleus, but monocyte has a dented nucleus. It looks like the nucleus was round but got a little dent. So it has a dented or kidney bean shaped nucleus. It's not bilobed. And from the blood monocytes comes the macrophages, and remember the macrophages, they perform phagocytosis, and that they perform phagocytosis, cleaning up all the debris found in the blood tissue. So we will talk about plasma first, and now we are switching gear and moving on to the PowerPoints. So plasma is a light straw-colored liquid, and it is about 55% of the blood volume, as we talked about. It contains about 7% plasma proteins, and we did mention a good friend remembers: albumin, globulin, fibrinogen, and regulatory proteins. Now, one thing that students sometimes ask me, Rupa, what is serum? As a healthcare professional, you will come across the term serum a lot. How is serum different from plasma? Well, serum is plasma minus clotting factors. So serum is plasma minus clotting factor. That's serum. Okay, I want you to remember this for testing purpose. So the first plasma protein is albumin, as we mentioned before. This is the smallest and the most abundant plasma protein. It maintains osmotic pressure and helps in water regulation in and out of blood vessels. Albumin also helps in transportation of some proteins, hormones, and small ions. That's albumin. And for these, just go by the PowerPoint. The next plasma protein is globulin. This is the second largest type of plasma protein, and about 37% of plasma protein is globulin. I forgot to mention about 58% of plasma protein is albumin, and 37% of plasma protein is globulin. There are three types of globulins: alpha, beta, and gamma. As I mentioned before in my drawing, alpha and beta, they help in transportation of lipids and other ions, and gamma globulin makes antibodies. Then there is fibrinogen. About 4% of plasma protein is fibrinogen, and that helps in blood clot formation. And then about 1% of plasma protein are regulatory proteins, and they just hang around as enzymes and hormones. And the easiest way to remember this, a good friend remember. So we have albumin, globulin, fibrinogen, and regulatory proteins. All right, moving on to the formed elements now. Remember, the formed elements have white blood cells or leukocytes. There are erythrocytes and platelets. I want to remind you that erythrocytes is about 44% of the total formed elements, and leukocytes and platelets together make up 1%. So we have about 45% of formed elements in blood. Rupa, do we need to know these percentages? The answer is yes. Okay. So let's start with erythrocytes here. And how much of your blood is erythrocytes? Well, that is measured by something called hematocrit. And hematocrit is defined as a percent of RBC in a total sample of blood. Therefore, let's say you take blood sample from a patient, you spin it down, and there are special chart-like things that are used to actually measure hematocrit. So with that, you can actually read the total volume of red blood cell. And it's usually the average in humans is 45%. That's the average hematocrit, meaning the percent of RBC found in blood. However, do note that females have slightly lower hematocrit value. It's between 38 to 46%, and males have slightly higher hematocrit value. It ranges between 42 and 56%. The reason being, we females, we menstruate, so we lose a little bit of blood every month. Therefore, we have a lower hematocrit value as compared to males. Here in this picture, you see a normal hematocrit where it's about 45% RBC in the total blood sample. If you are anemic, you will definitely have less RBC, less percent of red blood cell in the blood sample. And there is another condition called polycythemia where you have more red blood cells. The percent of red blood cells is higher in the blood sample. All right, talking about the structure of RBC. Now, I want you to look at the diagram here. RBC looks like a flattened disc, right? It doesn't look like a circular round cell that we draw out all the time. So RBC has this typical flattened shape. And remember, RBC does not have nucleus, so it lacks nuclei and other organelles. So mature RBC lacks nucleus and other organelles. And so if a cell is lacking nucleus and other organelles, and if there is damage to the cell, do you think the cell has the machinery to repair that damage? The answer is no. Therefore, if damaged, RBCs cannot repair themselves, and due to lack of this repair machinery, RBCs don't live long. They stay in circulation for only about 120 days. So the main function of RBC is to transport oxygen and carbon dioxide. In your exam, if I ask you, RBC transports oxygen only, is that true? No, that's false. Remember, RBC transports both oxygen and carbon dioxide, both. So, RBCs are loaded with hemoglobin. It's a pigment that is present on the surface of RBC. And in one tiny RBC or red blood cell, there are about 280 million hemoglobin molecules. And it is this hemoglobin that helps in oxygen and carbon dioxide transport. Now we are looking at each hemoglobin molecule here on the right hand side, and you see that each hemoglobin is made up of 4 globin chains. We have 2 alpha chains in green and 2 beta chains in yellow. And at the center of each globin, there is a little heme group in blue. Do you see the blue circle at the center of each globin protein? That's a heme group. And the red ball is iron binding there. And it is the iron where oxygen binds and is transported to different parts of the body. So to review, we are looking at each hemoglobin protein. Each hemoglobin is made up of 4 globin proteins, 2 alpha and 2 beta. At the center of each globin, there is a ring-like structure in blue. That's the heme part. And at the center, we have the red ball, and the red ball is iron that actually binds to oxygen and transports oxygen. Therefore, each hemoglobin molecule can bind about 4 oxygen or 4 carbon dioxide. Okay. Erythrocytes. Now, before I talk about the life cycle of erythrocyte, I want to tell you something. Now, when I was a student, my professor told me that we have about 5 million RBC per cubic millimeter of blood. How much? 5 million RBC per cubic millimeter of blood. I just memorized that number. But how much is actually 1 cubic millimeter? How much is that by volume? Well, please pay attention. If I put a small dot here in red, as you see, this is actually 1 cubic millimeter. Do you see that? This small dot, the small red dot, is 1 cubic millimeter. And can you imagine this 1 cubic millimeter of blood, or this one little dot of blood holding about 5 million RBCs? So you can actually imagine how tiny they are. They are super, super tiny. As I mentioned before, RBCs, they lack nucleus and no other organelle, so they lack everything. Therefore, in case there is damage, the RBCs cannot repair themselves. Therefore, that makes RBCs having very short lifespan of about 120 days, and they are in circulation for about 120 days. After that, they are broken down and recycled. So let's look into the life cycle of RBC. So the RBCs, they are born in the red bone marrow, and the RBCs, they float in circulation for about 120 days. And after 120 days, the RBCs are phagocytosed in liver and spleen, and a lot of the components of RBC are recycled. So let's talk about that. So after being phagocytosed in liver and spleen, the hemoglobin molecule splits up. And the heme part, remember the blue circle? The heme is actually converted to biliverdin first. So heme is converted to biliverdin first. And then the biliverdin then changes into bilirubin, which is actually a component of bile. So remember the heme part, or the blue ring that you saw in the hemoglobin, the heme gets converted into biliverdin, which eventually converts into bilirubin, and that forms bile pigments. What happens to the red balls? Remember the red balls, or iron? Well, iron is transported in blood by the protein transferrin. So there is a protein called transferrin, and this protein carries the iron to the liver. It carries the iron to liver, and in the liver, the iron is stored in another protein called ferritin. So let's review. The iron is transferred in blood by a protein called transferrin. So transferrin carries iron in blood. The transferrin takes the iron to the liver, and in the liver, the iron is stored in another protein called ferritin. So transferrin transports iron in blood, carrying it to liver, and in liver the iron is stored in another protein called ferritin, and this will be used later for producing new red blood cells. And a lot of the membrane proteins, they get dissociated, and the free amino acids are recycled back. All right, blood typing. So I have made a very, very detailed video on blood typing, very detailed, and you can actually expect some hard questions on your exam based on your understanding of the blood typing. You will get complicated questions where I will say, okay, this is the donor, this is the recipient, this is the donor's blood, this is the recipient's blood. Tell me whether the blood